I became interested in genealogy and genetic genealogy in 2014. At that time, I was very interested in finding the birthplace of one of my great grandmothers, and I'm happy to report that I eventually found her birth record in Brussels after a lot of work. So I've been doing genealogy for 9 years. Some people say you have to go farther back than that if you want to build a good tree. In 2015, I developed my first mathematical formula pertaining to DNA, and in 2019, I released my first genetic genealogy tool on the internet. And now I have a question to pose. A person is asking for help with a surprise DNA match. They share 1763 centimorgans of over 49 segments and can see that the match is on their paternal side. What's the relationship? I can tell you that this match is a paternal aunt or uncle. I can do that without knowing the ages of the testers, and this presentation will be mostly about how I can do that. But first, we have quite a bit of background to cover. I've been building relationship predictors for over 2 years. Traditionally, these tools have helped people by showing them all of the possible relationships for their DNA matches and probabilities for each relationship. You simply enter the number of centimorgans, commonly abbreviated CMs, that you share with a DNA match, like in this tool from DNA Painter. You can see every possible relationship and the abilities for each group of relationships below. This presentation is all about relationship predictions, so you might be surprised to hear that 3 years ago, I didn't see the need for them. I wondered why people wouldn't just look at the range of shared DNA for each relationship. You could even consider the differences in maternal and paternal relationships when doing so. Scientists have known for some time that paternal relationships are more variable because they have fewer recombinations on average, and so they have wider ranges. Maternal relationships share more segments and have narrower ranges. So 3 years ago, I was looking at tables like these instead of using relationship predictors. Here are just a few example relationships. This shows that the paternal half-sibling range is over 200 cms wider than the maternal half-sibling range. A match sharing below 1,300 cms has about a 0% chance of being a maternal half-sibling, but has some probability of being a paternal half-sibling. The reason I started building relationship predictors was that I realized I could provide a much more specific prediction than what was available to people at the time. Here's the probability curve that shows how a relationship predictor can work based on centimorgan values. On the x-axis, we have CMs, and on the y-axis, we have probabilities. The y-axis on the right is the same as the one on the left. The word relative here doesn't mean that these curves represent our relatives. It means that each probability for a relationship here is relative to the other relationships for a given CM value. That means that if we included more relationships, the probabilities would be lower, and if we included fewer relationships, they'd be higher. The probabilities only really have meaning in comparison to the other relationships. You might notice the abbreviation AVUNC for avuncular relationships here, which refers to aunt, uncle, niece, nephew relationships. Also, abbreviations like 1C stand for first cousin, and 1C1R is short for first cousin once removed. But the first thing that most people notice when they see this graph is that the grandparent-grandchild curve in green has two different peaks. Their initial reaction is that this must be a mistake, but it isn't. And I think it's absolutely beautiful. When I first saw the grandparent-grandchild curve, I had to think about it for a while. Eventually, I realized that at certain CM values, there are relationships that were much more probable than others, even though they share the same average. Since the grandparent-grandchild relationship is more variable, that is, it has a wider range, that relationship is more likely at CM values that are in between those of other groups. In this case, when the CM value is halfway between the average for full siblings in light blue and the close family group, that includes half siblings, the grandparent-grandchild probability is higher than that of other relationships. Since this is a graph for 23andMe data, and they show the full amount of full sibling sharing, the light blue curve is shifted more to the right, and that leaves a lot of extra room for grandparent-grandchild to have a high probability on the right side of its curve at higher CM values. The probabilities for each CM value have to add up to 100%. So when some relationships are less probable than other relationships, one relationship has to fill in the gap. There are CM values here where you could get a prediction with a probability of 80% for a grandparent-grandchild relationship. That really sets grandparent-grandchild apart from avuncular and half-sibling relationships because it's more than 4 times more probable at some CM values. It's really only the grandparent-grandchild differences that I want to concentrate on for this graph, although the rest of the curves are pretty interesting. There are also differences in probabilities between maternal and paternal relationships. Here are more specific probability curves just showing 4 close family relationships. I've left the avuncular relationships out because they have the most average curves and would clutter up the center of the graph. That means that the probabilities you see here don't add up to 100%. Again, we see the grandparent-grandchild curve sticking out with higher probabilities at the edges. But when we split up the relationship by maternal and paternal sides, it's the paternal-grandparent relationships that are really different. There are CM values where paternal grandparent in green is 3 times as probable as a maternal half-sibling in blue. You might be thinking relationships within groups are a little bit different and there are small differences between maternal and paternal relationships, but this won't result in more definitive relationship predictions most of the time. But just wait. Here's a screenshot of a prediction you can get from a tool at dnasci.com. A woman posted online that she shares 38.2% DNA with her paternal grandmother. She was told by several people that the probability of that relationship was 0 and that the amount of shared DNA was out of range. A woman is guaranteed to share an entire copy of the X chromosome with her paternal grandmother, which causes them to share 182 CMs more than some other relationships. The relationship is confirmed because the woman's father has tested and matches both women normally. This is a percentage rather than a CM value, but since those two metrics both tell us the amount of shared DNA, and since percentages at 23andMe include X DNA, you can get even better predictions from percentages. I know that isn't what most people have heard, but it's true and it's shown starkly here. Something I've learned repeatedly in the past couple of years is that more information is better. For this prediction, if you enter 38.2 in the percentage box for 23andMe and you click submit, you get the probabilities below. This is a high amount of shared DNA, but it isn't unreasonably high. About 1 out of every 5 matches who share this much DNA will be a paternal grandparent-grandchild pair. With the probability of the correct relationship only at 20%, it's still the only possible relationship in this case. Full siblings would share 2 copies of each chromosome over about a quarter of their genome, which would be clear to see in the chromosome browser. And they would have already been labeled as full siblings by 23andMe if that were the case. The lack of that type of sharing rules out full siblings. This woman knew the relationship, but she was confused when people told her that it wasn't possible based on other predictions. These predictions help in this case by taking into account the fact that grandparent-grandchild has a wide range of shared DNA, and also including the X DNA for 23andMe matches. And it's even more important to get this right when a person doesn't know who their match is. That last predictor you saw was called Origin. It was released in February of 2022 and was based only on CMs or percentages. Like all relationship predictors, the data that were used to generate probabilities came from simulations. Most of what people have used so far came from simulations conducted by Ancestry DNA in 2016. The DNA Painter tool we saw earlier simply uses a copy of the probabilities from Ancestry simulations. Most people who use the tool think that the probabilities came from empirical data related to a separate chart displayed below the probabilities, but there's actually a link right on the page that takes you to a blog post that describes how the data were copied from the Ancestry plot. Unfortunately, Ancestry and other matching sites have never described the methodologies they use for relationship predictors because they're proprietary. I've always included a lot of detail in my relationship prediction methodology articles, and last November saw the first ever publication of such a methodology in a science journal. There's the URL up there for the article I wrote, describing origin predictions in Forensic Science International. There's also a link to that paper on my About Me page at my site. So that's a brief overview of the developments in relationship predictions for the past couple of years. Finally, we can move on to what I really want to talk about. There's a new tool that's been available since February of this year, and it's much more accurate than origin predictions. It's called SegCM. And this is the mascot, created by my very talented wife and revealed today for the first time. Looks straight out of Sesame Street. But seriously, it's SEGCM, which stands for segments and CMs. This cluster diagram shows how the new predictor uses both CMs and the number of segments to show you how you're related to your DNA matches. It's CMs on the x-axis and the number of segments on the y-axis. On the right side, we have a tan cluster for parent-child matches. Then we have a yellow full sibling cluster that mostly stands apart from other relationships. In the center, we have a column of relationships that I most want to talk about. These are the 3 close family relationships: half-sibling, aunt, uncle, niece, nephew, and grandparent-grandchild. And each one has a maternal and a paternal cluster. So there are 6 total in that column. And on the left are patrilineal first cousins at the bottom and matrilineal first cousins at the top. It took a lot of work to find colors that didn't all turn into an indistinguishable brown in the center of the plot, and I've added some transparency so you can see through some of the clusters. This plot shows that for all relationship types, maternal ones share more segments on average, and so their clusters are higher on the plot than their paternal counterparts. When I first saw this plot, I didn't think that there was much difference between the clusters, and so I didn't think the relationship predictions would be that great. But boy, was I in for a surprise. The red cluster at the top is for maternal half-siblings, and this cluster is almost identical in overlap to the light blue cluster for maternal avuncular relationships, even though the colors make it easier to see the red half-sibling cluster. The pink cluster is for paternal avuncular relationships. It has a very similar shape to the light blue maternal avuncular relationship cluster. but the pink paternal avuncular cluster has a narrower CM range and has fewer segments, so it's lower on the graph. The purple paternal half-sibling cluster has significant overlap with the gray maternal grandparent-grandchild cluster. Finally, at the bottom, there's the blue paternal grandparent cluster, which is kind of similar to the purple paternal half-sibling cluster, but lower in number of segments. You might not be able to tell because of the transparency, but the paternal grandparent cluster has a significantly wider CM range than the other relationships. Let's look at how to use the seg-CM predictor. Where it really excels is for close family, so I want to show a few examples of those. Like with other predictors, you enter the total CMs into a box here at the top left labeled CMs. But now there's another input box to the right of that. There you enter the total number of autosomal segments. You can find that value very easily for all of your matches at 23andMe, Ancestry, or MyHeritage. You can also find it at GEDmatch in the one-to-one tool. After entering the number of segments, you click Submit or hit Enter, and you see all of the possible relationships and probabilities in the table below. I made up the numbers for this screenshot on the right. I was looking for a prediction that shows the 6 possibilities for the close family group, and that's something I rarely see. But I think it's useful when you're starting out to see which ones are possible. So we have 3 relationships listed, and each one has a maternal and a paternal option. There's no correct relationship here. What I want to illustrate is that splitting 100% into 3 relationships gives you about 33% for each one. If you include the paternal and maternal sides, then it's about 17% for each of the 6 possibilities. For a close family prediction, the best you can hope for is a 100% prediction like this one. Anything above 33% for the correct relationship type is a good prediction, and anything above 17% for a subtype is good. If we can't get averages that are higher than those values, then it would have been better to leave the relationships in groups and not try to point anybody to the relationship or subtype. So on the last slide, we had a 50% probability for a paternal half-sibling match, which was the correct relationship. And that's almost 3 times higher than the 17% that you would expect. Not long after seg-CM was released, a friend of mine plugged in the values for their paternal half-sibling. They didn't see the benefit of the tool in the results shown on the left. After all, the correct relationship was the second one down, and the third one listed by subtype. But that isn't how I use this tool. I look at the most likely subtypes. The first relationship I look at in this case is paternal half-sibling. And after that, it would be maternal grandparent-grandchild. I also try to find out whether the relationship is maternal or paternal first. And there are a lot fewer probabilities when I can do so. Another thing is that I'm used to skipping over grandparent-grandchild because it's often impossible based on ages. And back when I was using CM-only predictions, grandparent-grandchild was often the top prediction. So I got used to skipping over it often. The amazing thing here is that paternal half-sibling is the most probable subtype and the correct one. The close family relationships aren't even the only ones possible here, but the probability is still really high for paternal half-sibling. If I entered only CMs, the total close family probability would be about 90%. Dividing that by 6 subtypes yields about 15%, so anything better than 15% provides some value. If you can see the small screenshot on the right, the prediction from CMs only was about 18% for paternal half-sibling. So slightly better than 15%. The 44% from seg-CM on the left is much better. The answer to my friend is that they got a very good prediction, about 3 times better than if we used only CMs and left the relationships in groups. I've introduced something new in this screenshot. People often have questions about the paternal and maternal designations for the first cousin group. By the convention used here, a paternal maternal first cousin is your father's sister's child. A paternal maternal half aunt is your father's mother's child. And a paternal maternal great aunt is your father's mother's sibling. Why is there a ×2 listed beside those in the relationship predictor? If you added up all of the percentages in the probability column, you wouldn't get 100%. If you multiplied all the probabilities in those rows by 2, then you would get 100% when you added them up. If the display did that automatically, then those would usually overshadow the other 2 options. Only one of the 2, paternal maternal or maternal paternal, can never be true. So the goal is to find out which side the match is on and then use that information. For example, if I find out that a match is maternal, then I'm only concerned with the maternal-maternal or the maternal-paternal probabilities for the first cousin group, not the paternal-maternal probabilities. The prediction on the left here was for one of the beta testers for the seg-CM tool. When they entered 1674 CMs and 18 segments for the paternal grandparent, they thought it was the only option with 100% probability. Before seg-CM, nobody ever had gotten a 100% probability for one of the 6 close family relationships. The prediction on the right came up in a video from Family History Fanatics by Andy Lee. This was another prediction of 100% for the correct relationship, paternal grandparent. Of course, a genetic genealogist's work isn't finished right after looking at a relationship prediction. It's still good to verify the relationship, though through the shared matches of both testers, if possible. And of course, people want to know not only that a match is a paternal grandparent, but who that paternal grandparent is, and likely who their father is. After the DNA comes the genealogy. Here's something I see at least a few times every week. Someone has an unknown match, they know it's on their paternal side, and SegCM shows that the only paternal option is an aunt or uncle. In this case, a person had two matches. One ended up being their paternal aunt, and the other one their paternal uncle. We know that they aren't maternal aunts or uncles, and we know that they aren't maternal half-siblings because it's a paternal match. The screenshot on the left is the one I asked about at the beginning of this presentation. This is how SegCM can often give people only one option when they already know that the match is on their paternal side. One thing that can make this easier is that Ancestry now has the side view feature, which can split up your matches into parent 1 side and parent 2 side. Once you figure out which side is which, then you can label them appropriately. But many people already know which side their matches are on. Of course, these predictions always work if your match is a niece or nephew. But in that case, you might see the label both sides, or at the very least, your shared matches will indicate both sides. In that case, you might not know if a match is a child of your full brother or sister. The screenshots here highlight the difference between a very good prediction and a very useful prediction. The probabilities for the correct relationship weren't that high in this case, 21% or 28%, while a paternal grandparent prediction from SegCM often gives 80% and up to 100% for the correct relationship. But paternal aunt-uncle predictions are generally more useful because they so often leave only one possibility if you know the match is paternal. On the right is another case for a paternal match, and the only option was a paternal uncle. This newly found uncle happens to have a known maternal half-sister who also matched to the person I was helping. So she's a paternal-maternal half-aunt. You can see on the left that the relationship is listed at the top of the second group. It's the third highest probability on the page, and it's the second highest on the paternal side, which is the side we're considering. There are 9 other probabilities listed that are lower. So SegCM works quite well for matches in the first cousin group, which shares 12.5% DNA on average and includes half-aunts. We've seen that SegCM performs quite well for close family paternal relationships. And I showed on the plot of clusters earlier that maternal half-siblings and maternal avuncular relationships have almost identical clusters, making it harder for SegCM to tell the difference. That doesn't mean that you won't get a good prediction for maternal half-siblings or maternal avuncular relationships. I often see probabilities around 53% for those when they're the correct relationship, which is much better than you can get from CMs alone. But it isn't as useful if you see that more than one maternal relationship is possible. Confirming a relationship to an aunt or uncle often requires the match to respond to your messages. They're the ones who can differentiate between an avuncular or half-sibling relationship based on whether they see both sides or a maternal label. But even if your matches don't respond, you'll still get a very good prediction for maternal matches from SegCM. Here's a case where a woman didn't know her maternal grandparents. She recently found both of them on the same day. On the left, we see that grandparents is the only maternal option, with a 46.6% probability. On the right, the probability is even higher, 48.6%. Here's something I've seen a lot over the years. A person asks if their match might be a half-sibling, and based on something like the screenshot from DNA Painter on the lower left, people will say that the probability is 100%. I haven't been chiming in on that, but it really isn't a 100% probability for a half-sibling. It's a 100% probability for the entire group. There are 3 different relationships. That means that the half-sibling probability plus the grandparent-grandchild probability plus the avuncular probability all add up to 100%. If you split them evenly, which is the fairest thing we can do for DNA Painter, each gets about 33%. If we're trying to compare, and people have been trying to compare individual relationships to whole groups, we can't say that the one on the left assigned 100% probability to half-sibling, and the predictor on the right gave half-sibling a 46.8% probability. Both assign 100% to the whole close family group. The 46.8% on the right is comparable to 33% on the left, and if you compare the 46.3% for paternal half-sibling on the right, it has to be compared to about 17% on the lower left. I've shown quite a few examples of how accurate SegCM is for close family relationships, but it's also great for extended family. So let's look at some of those. Here's something pretty amazing. First cousins once removed and half-first cousins are usually thought to have the same properties of shared DNA because they both have an average shared DNA of 6.25%. But SegCM shows that they're quite different. The first 2 screenshots are for known first cousins once removed. In the first one, half-first cousins isn't even the second probability listed. It's way down here below 3 relationships in the first cousin group. The correct relationship is 24 times more likely than half first cousins in this case. In the second screenshot, the correct relationship has an 87.7% probability. It's about 18 times more probable than half first cousin, resulting in an unnecessarily ambiguous prediction. First cousin once removed is where a person's eyes should go when they're trying to figure out a mystery match in this case. I've seen a lot of first cousin once removed predictions with probabilities in the 70s and 80s of percents. In the third screenshot, a half first cousin is the known relationship, and it's twice as likely as a first cousin once removed. There are other relationships listed along with half first cousin, like second great avuncular, but they would typically have a much larger age difference. Roberta Estes recently wrote a blog post about relationship predictions. Sexium wasn't mentioned in the post, but it showed the amount of shared DNA for 19 matches, including the number of segments. This provided an opportunity to compare Sexium to ancestry predictions. While empirical DNA match data tend to be very messy, Roberta likely vetted the data very carefully before publishing her work. In the first column is the known relationship. In the second and third columns are the amount of DNA shared. The last three columns show the probability that predictors gave to the correct group. The first one is for Sexium. The next column is labeled origin. It's the name of the predecessor to Sexium, and it's the probabilities that Sexium will fall back on if you don't enter the number of segments. You can also see that the probabilities are the same for some of the second cousins and more distant because of the number of segments doesn't help as the CMs get lower. And the last column is from DNA Painter, which uses the ancestry probabilities. The group for first cousins include relationships like half-niece shown here, also great avuncular and great-grandparent, grandchild. The group that includes first cousins once removed also includes half first cousins. The best prediction is shown in green for each relationship, the second best in yellow, and the lowest prediction in red. Ties for first are both shown in green, and ties for second are both shown in yellow. Sexium gave the highest probability of the correct relationship 63% of the time, all these green ones, leaving the other two predictors to split the remaining 37%. Origin usually had the second highest probability, 73% of the time, all those yellows. And ancestry simulated probabilities had the lowest probability for the correct relationship, 68% of the time, the reds there. For the average probability assigned to the correct group, shown in the bottom row, predictors tend to perform similarly. The origin predictor did decently better than the ancestry probabilities, but Sexium only had 1 percentage point higher than origin. This is in line with what we've already seen, that the great advantage of the number of segments is at differentiating between relationships in the same group. So let's look at individual relationships, which are now shown to the left of the group probability columns. Unfortunately, the maternal and paternal sides weren't listed for the first cousin group, so I had used the total probabilities for first cousin, half-niece, etc. Sexium gave the highest probability to the correct relationship 50% of the time, over twice as often as the ancestry probabilities. This time, origin almost always had the second highest probability, 90% of the time, which shows that CMs alone have some ability to distinguish between groups. And ancestry simulated probabilities had the lowest probability 68% of the time, which was the same as for the group probabilities. But now we actually see a real difference for the average probabilities assigned to the correct group, shown in the bottom row. This time, Sexium did much better than the ancestry probabilities, but the origin probabilities did only marginally better than ancestry. You can see that when the ancestry probabilities did better than Sexium, the differences were typically very small. The highest individual probability from ancestry simulations is only 50%. It's definitely a good idea to use Sexium because it assigns 76, 73%, 78%, 71% to the correct individual relationship. Let's look at this prediction that gives 78% to a second cousin when it's the correct relationship. Two people from Roberta's dataset shared 213 CMs and 9 segments with each other. If they didn't know the correct relationship, this would have shown them a pretty low probability for any relationship other than the second cousin group, which gives a person a great place to start. Predictors used to struggle with relationships like second cousins and more distant. DNA Painter only gave the second cousin group 49% in this case, and Origin gave it 68%. This isn't just a one-off example. 49% and 50% were the highest probabilities that DNA Painter gave to a second cousin or more distant in Roberta's entire data set. The more data we look at, the clearer it is that Sexium gives very good predictions for any degree of relationship. So far, I've only talked about ancestry and DNA psi predictions today. While I was at RootsTech earlier this year, I saw that MyHeritage announced a new predictor that takes ages into account. That really caught my attention because I had just released Sexium a few weeks earlier. This screenshot shows a made-up close family prediction, and I haven't entered ages yet. It shows that the close family group is broken into 5 relationships at MyHeritage. because that's the best way to take ages into account. Let's look at how we would compare this to other predictors. I checked, and there doesn't appear to have been a rigorous comparison of MyHeritage probabilities to any other predictors yet. If one really wants to see if including ages adds any value, and they want to compare to a predictor that includes groups, there's only one good way I can think of doing that. For the individual predictor that only includes groups, they'd have to divide the close family group into 3. So for a prediction of 100%, each relationship would get 33%. Then, since MyHeritage shows aunt-uncle separately from niece-nephew, the 33% would again be divided by 2. The same would be done for grandparent-grandchild. So for a prediction of 100% close family group, half-sibling would get 33%, but aunt-uncle, grandparent, and grandchild would each get 17%. These values in pink show the probabilities that MyHeritage has to beat in order to add value by including ages, or the ways to split up groups from other predictors when comparing to MyHeritage. For other relationships, such as first cousins once removed, MyHeritage reports the probabilities separately for the younger generation and the older generation. One would either have to add those probabilities together, along with half first cousins, in order to compare probabilities to a predictor that lists all of those in the same group. Or preferably, they'd have to divide the probabilities from the predictor that uses groups according to how they're split up at MyHeritage. I know dividing the group probabilities by the number of relationships isn't intuitive to a lot of people, and there are even people who just wouldn't have thought of doing it, but we can only compare like to like. Out of my nieces and nephews, only one has had their DNA tested. MyHeritage gives us the highest possible prediction for the correct relationship, 100%. SegCM also gives a very good prediction for this relationship subtype, 42.6%, that I'm his maternal uncle, with the only other reasonable option being maternal half-sibling. Neither of these values is at the resolution we need to compare the two. The MyHeritage predictor didn't specify if this was a maternal or paternal match. We'll have to split those evenly with 50% each. SegCM didn't specify if my match was in the generation older or younger than me, so I'll have to split the 42.6 into 2. We should always use the finest resolution subtype that allows us to compare like to like. Otherwise, we don't know if the predictors are adding value by showing finer resolution. The final result is that SegCM assigned 21.3% to the finest resolution subtype, and MyHeritage assigned 50%. The MyHeritage predictor has done very well here. My family does have half-sibling pairs with similar age differences, but MyHeritage is telling us that that will happen fewer than 1 out of 1,000 times, resulting in a 0% probability for half-siblings. Some might say that there's no such thing as a maternal nephew because a nephew is related through both sides. I agree that a nephew is related through both sides, but because of a lack of any other term that works, for my purposes, a maternal nephew is a nephew to whom I'm a maternal uncle. I have many half-siblings, and all of my maternal siblings have tested. In my family, there are maternal half-siblings who are 22 years apart. Choosing that pair would be pretty unfair to the MyHeritage predictor, so I use the CMs between myself and the one who's closest to my age. The MyHeritage predictor did great. Other predictors would give less than 33% to half-siblings based only on CMs. If we're not comparing subtypes, the MyHeritage predictor beat SegCM 86.2 to 50%. But MyHeritage is lumping maternal and paternal half-siblings into one. In this case, MyHeritage assigned 43.1% probability to maternal half-siblings, and it was 50% for SegCM. That's very close, and they're doing different things, so they both add value. Now to compare MyHeritage and the 2016 ancestry probabilities for my same half-sibling. We don't need to split the probabilities for paternal and maternal sides this time because neither predictor shows that, and half-siblings only have one perspective. MyHeritage gave the relationship 86.2% probability. DNA Painter has rounded the value from 99.72% up to 100%. Keeping the unrounded value and dividing by 3 yields about 33.2%. The MyHeritage predictor is over twice as good in this case, and these are very typical results. One thing we definitely can't do is compare the probability of a half-sibling at MyHeritage to the entire close family group at DNA Painter. That would allow DNA Painter to work with a total of 300%, while MyHeritage is stuck with the normal total of 100%. What I mean is, if DNA Painter gave half-siblings a 100% probability in this case, then it's also giving grandparent-grandchild a 100% probability and another 100% for avuncular relationships. But a 300% probability is impossible. Of course, the probability for half-siblings can't be higher than the whole close family group that includes half-siblings. A prediction from MyHeritage or SegCM for half-siblings will usually be much higher than from ancestry simulations, but only if we compare half-siblings from one tool to half-siblings of another tool, and not the whole group. There's no question that MyHeritage predictor adds value when it takes ages into account, rather than using only CNs. I do have to wonder, though, if the genealogist is best equipped to take into account the ages of the testers, along with all the other demographic information, such as ages of other people in the family tree and places where they lived. Ages can sometimes be deceiving. Another measure of a relationship predictor is how often it gives 0% probability to the correct relationship. That's something that will happen with every predictor. We saw earlier the woman who shared 38.2% with her paternal grandmother. The tool at DNA Painter said that that had a 0% probability based on ancestry simulations, and that it was out of range based on empirical data. But the MyHeritage predictor seems to give 0% to the correct relationship more often than others. That puts them in a tricky position. If their input data are correct, then they're simply reporting the true probabilities. One solution would be for them to give less weight to ages, but then the predictions wouldn't be as strong most of the time for good predictions. I think it can be a good idea to look at the predictions based on ages. It's especially useful if the DNA sites know and use the exact ages of the testers, but they're hidden from or approximated for their matches. A little while after RootsTech, MyHeritage announced that they had been working on an algorithm that takes both ages and the number of segments into account, and said that it works phenomenally, which isn't surprising at all. Although they didn't mention that SexCM was already using the number of segments. There's one more major feature of DNA-SCI predictions that I haven't discussed yet, and that's population weights. Some people call it a population growth model, but the principles remain true whether the population is declining or growing. It's a well-known fact that your distant cousins far outnumber your cousins from just a couple of generations more recent. Because of that, your distant cousins are far more likely to show up in your match lists, even if you sort by highest CMs. We need to account for that in relationship predictions. I find that it's easiest to understand it by choosing a survival rate and assuming that it's the same for every family in a population. It's probably easiest to pick 2 surviving children per family. In that case, each person would have, or 1 sibling. You would also have 1 aunt or uncle on each side of your family, so 2 in total. In turn, they would each have 2 children, leading to a total of 4 first cousins. How do we count more distant cousins? You'll have 4 pairs of great-grandparents. Each pair will have 1 child who isn't your grandparent. You can multiply that 4 by 2 to get your first cousins once removed from the generation older than you, and multiply again by 2 to get 16 second cousins. This is how I developed formulas to use for origin, only I did it the hard way with a survival rate of 2.5, which is that SR there. I observed the patterns and wrote a formula. I also developed a formula for cousins once removed because relationship predictors include those. So we can't just have first, second, third cousins, etc. We need the groups halfway in between each of those. After all of that, I realized that some scientists from 23andMe had already developed the formula for cousins not removed. Once you have the numbers, the probabilities for a relationship are increased according to its weight, giving higher probabilities for distant relationships. Each generation has about 5 times as many cousins as the previous if you use the survival rate of 2.5. If you randomly chose a DNA match, they're 5 times as likely to be a second cousin as a first cousin. If you picked a DNA match that had equal probability of being a first or second cousin in an unweighted predictor, that match is 5 times more likely to be a second cousin than a first cousin, and a weighted predictor will show that. As we go back further, at some point our distant cousins won't grow at the same rate because of pedigree collapse. That's something that will hopefully be taken into account in the future. What's shown in this table won't be the exact number of cousins that you have. What's important is that this approximates the population on average. I'm sure it could be improved upon by having different numbers for different populations, but so far nobody's done that, and it's much better to use those population weights than none at all. Predictions at DNA-SCI use the peer-reviewed population weights of the Hen et al. 2012 paper, and that makes them more accurate for distant relationships. All of the screenshots shown here are for HCM matches, but from different tools. The prediction on the left is for population-weighted predictions at DNA-SCI, which properly show that the match is likely very distant. In fact, these predictions only go back to 8th cousins once removed, so the most likely relationship might be even more distant. This is good information to have. Some people will decide not to investigate an HCM match. Others will have a better idea of where to look for one. The next one shows unweighted predictions at DNA-SCI. These are the only predictors for which the developer, yours truly, is on record stating whether or not population weights were used, and that's the only difference between the 2 tools. Not shown are the ancestry probabilities from 2016, which are rumored to have included population weights, and that appears to be true. The third screenshot is from the 2019 ancestry probabilities, which are used in what's called the DNA Painter beta tool. These are almost identical to the known unweighted predictions from DNA-SCI in group order and probability. There's no question that they left out the weight since 2019. The fourth screenshot is modified from MyHeritage to fit on the screen. It's much more similar to unweighted predictions than to weighted. If they used population weights for the predictor, They're very, very mild weights. Population weights bring up another complication when comparing relationship predictors. Comparisons are done using known relationships, but you have to make sure a dataset has more cousins with each generation and in the right proportions. Unfortunately, empirical datasets tend to have fewer relationships with each generation once you get past third or fourth cousins. This is the opposite of what you'd want. Unweighted predictors have an advantage over weighted predictors unless you ensure that there are more cousins with each generation and in the right proportion. Remember, the purpose of a relationship predictor is to find out how we're related to unknown matches. One solution is to only compare predictors with population weights to other predictors with population weights, which is what I've always done. That makes it difficult because it appears that the only tools with population weights are the Ancestry simulated data from 2016, which are used in the original DNA Painter tool, and predictions at DNAsci. It appears that MyHeritage doesn't use them, and Ancestry dropped the population weights in 2019 to present. I've just analyzed 209 data points this week, and these are the results. This table shows the known relationship of DNA matches. The next column is the best possible prediction that you could get for an individual relationship if they're lumped into groups, and that group gets 100% probability. The next column shows the SegCM probability, then the DNA Painter probability, and then the improvement of SegCM over DNA Painter. All probabilities are average individual subtype probabilities, even for the first cousin group relationships. There just isn't room to show all 16 of the relationship subtypes for the first cousin group. It would not be interesting to show the group probabilities here. SegCM gets 99%, Origin 98%, and DNA Painter 97% for close family relationships. So every predictor is pretty good at predicting groups. The DNA Painter probabilities do well here, almost keeping up with the best possible from groups column. But for all these relationships, SegCM provides an improvement over that. For most, the improvement is substantial, and the paternal grandparent-grandchild probability stands out as usual. On average, when a person enters the CMs and segments for an unknown paternal grandparent-grandchild relationship, they'll get a 96% probability for the correct relationship. From the close family to the first cousin group, SegCM gets double the expected probabilities or more. I intend to evaluate progressively larger data sets. If you follow the link on the page, you can find a link to a survey for DNA match data that will be very beneficial for the new scientific discoveries or tools. In the words of my favorite 80s robot, need input Stephanie. I also have a set of over several thousand data points. I've employed a 2D statistical outlier detection algorithm that requires no subjective input for each relationship, and it works great. I also had to write a long algorithm to standardize the relationship labels for that data set, but now that it's done, it shouldn't be long before I'll have results from that data set of several thousand too. From the MyHeritage predictor, we've seen that ages improve the probabilities given to the correct relationship. I don't think we should always look at predictions that take ages into account. Sometimes it's best if we consider the ages on our own, but it usually won't hurt to take a look at those predictions. The number of segments, on the other hand, is something we should always consider. We're not good at guessing how probable a relationship is based on CMs or segments, but we're very good at taking ages into account. For now, the most accurate and helpful predictions that you can get are from SegCM. If there's a problem with entering the number of segments or the results you get from that, the next best predictions you can get are from 23andMe Percentage Box, also at SegCM. And you can get the third best predictions by entering the CMs into SegCM without any segments. Also, some skepticism about proprietary company information is justified. Ancestry have told us very little about the simulations that generated their probabilities. And I don't think that MyHeritage has said anything about where their centimorgan data came from, although they showed some plots of age distributions that presumably were part of their calculations. SegCM and Origin are trustworthy sources of predictions because the methodologies are described clearly and in detail. There are many people who have never found out how they're related to their DNA match. Some of them gave up years ago. A lot of the time it's because their match doesn't have a tree, doesn't have a full name listed, and doesn't respond to messages. People don't have to give up anymore. With SegCM, a person can see that the only maternal option is that their match is a maternal grandparent, or they can see that the only option at all is a paternal grandparent, or they can see that the only paternal option is a paternal aunt or uncle. And in a lot of other cases, they'll get very high probabilities for one option over the others. I sincerely believe that the single most important thing I can do as a genetic genealogist, rather than building or improving upon tools, is to spread the word that people can now find out how they're related to their DNA matches. Not that you won't see any more tools from me. I already have plans for some more great tools. Thank you so much for your time, everyone.